Hello students and welcome to Malhotra's IAS Academy. I'm Surbhi Sardana and today we are going to study genome sequencing and the Genome India project. The Genome India project has been in news recently. We'll see why this has been in news. Apart from this, we'll also learn what genome sequencing is, what is Department of Biotechnology doing with regards to genome sequencing, and why genome sequencing or the Genome India project is important for our country. So let's begin. Let's see what the news is about, what has been coming up in the news, and that which makes this topic so important. First of all, recently the Department of Biotechnology, it has said that the exercise to sequence 10,000 Indian human genomes is about 2/3 complete. That is 75% human genomes of India, they have been sequenced. About 7,000 Indian genomes have already been sequenced, of which 3,000 are available for public access by researchers. So sequencing or genome sequencing of 7,000 people has been done. And this exercise basically it is being conducted to create a database under the center backed or the central government's project, which is also known as Genome India project. This Genome India project is is a project under Department of Biotechnology. A lot of research institutes are involved, a lot of labs are involved for this project all over the country. So see, Genome India project is something which is very important for your prelims, mains, interview, all stages of the exam. But for that too, for understanding what Genome India project is about, the first basic clarity of any concept that is required here is understanding the genome. So what is genome? What is human genome? See, to give you a basic overview, आपको एक बहुत ही simple तरीके से बताने के लिए, आपके जो inherited traits होते हैं, whatever inherited traits you have in your body, whether it's about your height, it's about your eye color, any kind of disease you might have in future, that is coming from your parents, that is from the father and from the mother. So half of your genes or half of the chromosomes come in your body from your father and the other half comes from the mother. So we'll look at a detailed picture of this. Now understanding or reading all these genomes, all these gene sequences is known as genome sequencing. So yes, what is a genome? Where is it present? For before reading, I want you to take you to this picture which shows you where the genes are present. So look at this picture, this part here, this figure here. This is one cell of your body. Cell is the fundamental unit of life in all living beings. So in living beings, for example, your body also, the smallest unit of life is the cell. Now each cell, just like every human being has a brain, each cell has a nucleus, that is known as the control center of the cell. So inside the cell, there is this nucleus and it is a very protected organelle. Nucleus बहुत ज़्यादा protected होता है. It is protected by nuclear membrane. So nucleus is there and inside the nucleus you have chromosomes. These X-shaped things or these tube-shaped things which you see here, which has been marked also in the circle, these are chromosomes. Now all of us, all the living organisms, in the start of our life, in the beginning of our life, we just started our life from one cell. We were one cell which multiplied, multiplied, got divided, and hence a human being or a living organism comes out of a mother's womb, that way, in case of mammals. So yes, you have the cell here, you have the nucleus here. So we'll be studying just one fundamental unit of life, the cell. Inside the nucleus you have these chromosomes. Now chromosomes, in human beings we have 46 chromosomes. 23 chromosomes, they come from your mother and 23 chromosomes, they come from your father. So these chromosomes, these are to be studied because chromosomes have DNA on them. Now let me expand this picture here. This is your X-shaped chromosome. This is your X-shaped chromosome which we have taken out of the nucleus. On this X-shaped chromosome, there is a coil of DNA. DNA is coiled on these X-shaped chromosomes. And when you open this DNA, this is how it looks like. Let me give you a pictorial representation, a different pictorial representation. So let's say this is your cell. This is your cell. Inside the cell, you will have the nucleus. Inside the nucleus, you will have some like 46 chromosomes. And chromosomes are responsible for the genetic characteristics, जो कि आप inherited characteristics आप लेते हैं अपने parents से. So let's say now we take one chromosome. We have taken one chromosome out of here. Now what we have seen is that on these chromosomes, DNA is coiled like a wire. So this black colored coil on the chromosomes is known as DNA, that is deoxyribonucleic acid. और DNA क्या contain करता है? सारी information. DNA contains information of inheritance that is coming from your parents. So let's say we open this DNA up. तो आपको जो ये DNA अगर आप open up करके देखते हैं, it should stretch like a wire, it should look like this, but it does not look like a straight wire. Instead what it looks like is double helical structure. This is the structure of DNA, that is how it looks like. Now I'll take you back to the picture. When you have on the chromosome, you have DNA coiled on this. When you open the DNA up, this is how the DNA looks like, double helical structure. Now DNA has some bases or some base pairs. Bases क्या होते हैं? Bases are some chemicals basically. There are 4 chemicals which are present on inside DNA, that is adenine, cytosine, thymine, guanine. Now ACTG. I want you to remember that A forms a bond with T and G forms a bond with C. All these colorful things that you see here, these are basically base pairs. Base pairs के bases में जो bonds है, these are what we are seeing here. So yes, look in here. Adenine is in yellow color. Yellow or A is forming a bond with T. G, that is red in color, is forming a bond with C. Now these are the configuration of bonds. This is how bonding happens. Now let's say this is one bond of AT, adenine and thymine. Let's say this comes first and the second bond is of GC, that comes second. Let's say GC comes again or AT comes again. Let's say ET comes 10 times or GC does not come at all. So that is what? That is the change in the base pair, how base pairs are occurring inside a DNA. For example, if I talk about here, if we go back to this DNA sequence here, this DNA sequence is consisting of base pairs AT, GC, AT, and it could be like all this strand is formed up of AT, AT, and downwards it is formed of GC. So basically the configuration of these base pairs can be any, it can have any configuration. Now the configuration of these base pairs, it is mostly same in all the human beings. सारे human beings में जो configuration है mostly का mostly same रहता है. But there is 0.3% changes, 0.3 or 0.1% change in one person to another, change or difference. Let's say the difference in your DNA and your siblings' DNA would be 0.1%, or the difference in your DNA and your teacher's DNA would just be 0.1%. And how can you figure that out? When you study this DNA, when you decoil the DNA, when you study the configuration of base pairs on DNA. If you are studying the configuration of base pairs on DNA, that is known as genome sequencing, that is known as DNA fingerprinting, genome sequencing, whatever you want to call it. So that is what we are trying to do here. DNA का कुछ part होता है. Let's say this much length of DNA which gives you brown eyes. Let's say this much helix, this much double helical strand of DNA, or this much length of DNA which is responsible for giving you brown eyes or black hair. The minimum length of DNA that can give you one feature, whether it's hair, whether it's skin color, whether it's eyes, that is known as gene. So basically if you have a very long strand of DNA, you will have all the genes on it. Let's say this is here we have a very long strand of DNA. Look at this, this is a very long strand of DNA. If I say that if you take DNA from this point to this point, whatever base pairs or bonds are there between bases AT and GC, they will give you one feature, whether it's your eyes, whether it's your skin color, whether it is a particular disease. So this shortest unit or the fundamental unit of inheritance is known as gene. If you are studying all these genes and if you are studying the configuration of base pairs inside these genes, then that is known as genome sequencing. अभी genome sequencing का हमें फायदा क्या होता है? First of all, every organism's genetic code is contained in its deoxyribonucleic acid, DNA, the building blocks of life, just as we saw. A genome is all the genetic material in an organism. If you study all the genes, that is the genome. It is defined as an organism's complete set of DNA. So, DNA does not have to be cut from anywhere, it does not have to be present in parts. The complete set of DNA would constitute the genome, including all of its genes. Each genome contains all of the information needed to build and maintain that organism. All the features that you have in your body, in fact even your immunity, that is coming from the genes which are present, that is parts of DNA that are present in your chromosomes. In humans, a copy of the entire genome contains more than 3 billion DNA base pairs. So, the AT and GC base pairs that we were talking about, there are more than 3 million base pairs in human beings, in the entire genome. So, just imagine studying the entire genome, studying the configuration of all the base pairs, 3 million base pairs for a human being. That would be a humongous task, that would be a very big task, and these kind of studies have been carried out in the past, and that is what we are doing, that is what we are trying to do in the Genome India project. So, yes, the DNA consists of double-stranded molecule, just like I showed it to you. That is how these are double strands, one strand and two strands, built up by 4 bases: adenine, cytosine, guanine and thymine. Your A will bind with T and C will bind with G. In all, the genome is made up of approximately 3.05 billion such base pairs. So, these are the pictures that we have already seen. You have the cell, inside the cell you have the nucleus, inside the nucleus you will have these chromosomes. On the chromosome you will have this coiled DNA. When you open that up and when you study the configuration of AT and GC, which one is coming first, which one is coming second, which one is coming last, that will be known as studying the DNA, that will be known as genome sequencing. So, yes, just look at the picture of genome sequencing here. Here you have one gene, or what is one gene? Smallest length of DNA or smallest unit of DNA that can give you one feature. So, here when you carry that out, when you study the configuration of base pairs and you establish an information file in your lab, that is known as genome sequencing. So, while the sequence or order of base pairs is identical in all humans, in fact in many living organisms it is almost the same, but compared to there are differences in the genome of every human being that makes them unique. So, just like you have fingerprints which are different for every individual apart from twins, if you can study DNA of people, the configuration of base pairs in the DNA of people, that will be different for all human beings. And on the basis of that study, on the basis of studying the DNA or the genome of individuals, you can carry out various tasks. बहुत सारी applications हैं, there are a lot of applications and a lot of uses. That's why this area of research is coming up. So, the process of deciphering the order of base pairs to decode the genetic fingerprint of a human is called genome sequencing, just like we studied. In other words, genome sequencing is the process of determining the complete DNA sequence of an organism's genome. So, yes, now moving on to the Human Genome Project. We will also come to the Genome India project, but what is the Human Genome Project? See, back in 1995, there were a few scientists, कुछ scientists थे जिन्होंने कहा कि हम सारे human beings का या कुछ human beings का, let's study the genome, let's carry out genome sequencing of some human beings, and that should serve as a basis for us for studying other human beings. So, Human Genome Project was carried out, it completed in 2023 and the latest data has come to us in 2023. It completed in 2003, back in 2003, and the latest data has come up in 2023. So, the process of whole genome sequencing made possible by Human Genome Project now facilitates the reading of a person's individual genome to identify difference from the average human genome. Let's say you have results of Human Genome Project. So, you know how a human being's DNA or how a human being's gene looks like. A normal human being's gene, how does it look like? Now, there is a person who has some diseases that is coming because of some genetic issues. So, if you carry out studies or genome sequencing of this person who has a disease and you compare it with the results of Human Genome Project, so you can figure out what is the gene that is creating problem. You can figure out new types of diseases also just by genome sequencing of other individuals which might have a disease. So basically different type of medicines, targeted drug delivery or identification of diseases, identification of criminals. There are a lot of applications which have been made possible by the Human Genome Project. Now the problem with Human Genome Project is that yes, it has carried out sequencing for a lot of human beings, and it has given us a normal picture of how human DNA or how genetic genes of human beings look like. But the problem here is that who were the people whose DNA was taken to carry out genome sequencing in Human Genome Project? Who were these people? So HGP or Human Genome Project has a major diversity problem. These were not people from India, or these were not people from poor backgrounds, or let's say tribal communities which are prevalent in India. We have a lot of tribes in India, we have a lot of poor people in India. In fact, in India you can see much ethnic diversity than the rest of the world. If you go to South India, you find different kind of people. If you go to Northeast, the ethnic diversity is different. In Northern India, people have different kind of features and hence different kind of diseases as well. So Human Genome Project, the problem is the problem of lack of diversity here. As most of the genomes, over 95% people's genomes, the people who were taken for genome mapping, they were actually urban middle class white people. So brown people or black people, they were not taken, and these are mostly urban middle class white people. So Human Genome Project, the observations of Human Genome Project does not serve the interest of India, because our ethnicity, our DNA, that is very much different as compared to white people of developed countries or of Western nations. So now Department of Biotechnology under Ministry of Science and Technology, they decided that just like this Human Genome Project, we will have a Genome India project. We will have a Genome India project for our country, and we ourselves in India itself, we will carry out genome sequencing, where we will take some of the human beings, some of the individuals from all ethnicities, whether it is South, whether it's Northeast, whether it's Central India, North India, Western India, whether it's the tribal groups. So that is what your Genome India project is about. One thing to remember here is that genome sequencing of the entire 1.3 billion people of India, that will not be carried out. They will be carrying out genome sequencing of just 10,000 people. 10,000 people, and as the results have come up, they have told us that out of these 10,000 people, 7,000 genome sequencing for 7,000 people is complete. That is 2/3 of Genome India project is already complete. So the Genome India project, it is a gene mapping project sanctioned by Department of Biotechnology. It was launched with the goal of creating a comprehensive database of genetic variations among the Indian population. It aims to sequence the genomes of over 10,000 Indians from different regions of the country. So that is not just from one state, and all the people are not included, just a bunch of people, so that to include all the types of ethnicities they are taken for and establish a reference genome for the Indian population. So yes, these are the basic, first of all, just understand that this is an initiative of Department of Biotechnology under Ministry of Science and Technology. So what they are trying to do is cataloging the genetic variation in Indians. Reference genome for India, that will be prepared just like your Human Genome Project. It tries to establish a reference genome for the entire world, but it does not suit Indian needs. So reference genome for India, a national genome sequencing project to sequence 10,000 Indians, that will happen. Capacity building and networking, that includes training manpower to handle the large genetic data that will come up, because you will be storing it in your labs. And other resources across institutions, resources for larger and cheaper genetic studies, because obviously if you have 3 billion base pairs, 3 million base pairs in every human being, so storing that kind of data, that requires a lot of money as well. What else do we have here? Disease gene identification. So the type of diseases or the types of genetic diseases that are prevalent in India, the same types of diseases will not be prevalent in white population, because our genes are very much different. So disease gene identification, what are the genes? Some genes give you eye color, some genes give you straight hair or the color of your hair or the color of your skin. There are some genes which give you diseases as well. So those genes have to be identified. That is one of the features of Genome India project. Catalog all the genetic variations in Indians to help identify disease causing genes. Genetic basis for adverse effects and response to drugs. See, all the medicines that we use in our country, they are coming either from foreign countries, or even if they are produced in India, they are basically generic versions of medicines that are coming from developed countries. So these medicines which are coming from developed countries, they are prepared for the genetic variation or for the people staying in developed countries. They do not suit Indians, and even if they suit Indians, probably they they help us fight fever, they help us fight some infection. There could be n number of side effects they could be producing because of our genetic variations, because of the difference in our genes as compared to white people. So yes, genetic basis for adverse effects and responses to drugs, this will be basically one of the aims. Now, it allows researchers to learn about genetic variants unique to India's population and population groups, and use that to customize drugs and therapies, so that Indian population or Indian genes based drugs and medicines and medical treatments, they can be prepared. So for that to be happening, some data is required, and that will be coming from Genome India project. So that is going to be a major breakthrough in medicine that we will be seeing in the coming years. So India's 1.3 billion strong population, it consists of over 4,600 population groups. Just imagine 4,600 population groups, many of which are endogamous, that is, they marry within their groups itself. So first of all, there is a horizontal diversity that you have in India. Horizontal diversity is, let's say, in any part of India, we have people who belong to that land, and we have people who migrated from other countries or from other states. So that is the horizontal diversity. Secondly, you also have the vertical diversity, because what kind of marriages are happening in these population groups? Endogamy is prevalent, that they do not marry outside their caste, their tribe, their religion, or things like that. So basically, mutations that are introduced in the forthcoming generations, these are prevalent in these population groups only. So it is not a far-fetched reality that in the coming times, diseases on the basis of religion or any kind of occupation, that could be told to you in advance, that yes, or wherever you stay. Let's say if you stay in North India, if Genome India project it becomes successful, few years from now or few decades from now, the doctors will be able to tell that if you stay in North India, these are the diseases which you are likely to develop in your 30s, 40s or 50s. So that is going to aid the medical industry or the healthcare system a lot. Now finally, distinct variations with disease causing mutations are often amplified with some of these age groups, because they carry out endogamy. They do not marry outside their groups. So yes, now what can be the applications of this Genome India project? See, applications we've seen, first of all, disease diagnosis can happen at an early stage if you know which is the gene that can possibly cause a disease. Personalized medicine can happen because for different kind of population groups we can have different types of medicines. New drug development, new types of medicines can be developed in India which suit Indian population more as compared to the medicines that we have taken formulation of from developed countries. Then agriculture. In agriculture, obviously genome sequencing is important for your plant varieties as well. So in agriculture also there will be a lot of help. In forensics, identifying criminals. Secondly, identifying missing persons and victims of disaster. There are n number of possibilities that can happen once the Genome India project is successful. So let's solve a question here. This is one of the questions that was seen in 2017 in UPSC prelims. With reference to agriculture in India, how can the technique of genome sequencing, when you carry out genome sequencing with respect to agriculture, what you are doing is you are studying the DNA of plants. So with reference to agriculture in India, how can the technique of genome sequencing, often seen in the news, be used in the immediate future? So let's say if you study the DNA of plants, if you study which gene of plant is responsible for higher yield, or which gene of plant is giving the flowers a red color. So what are the possibilities that can be accrued? The first option is genome sequencing can be used to identify genetic markers for disease resistance and drought tolerance in various crop plants. In fact, if you talk about BT cotton and other genetically modified crops, this is the application that has already been developed there. Secondly, this technique helps in reducing the time required to develop new varieties of crop plants. That can happen if you can, carry out some genetic editing. If you know the genome, if you know the gene that is responsible for the yield, and if you can carry out some genetic editing, this can be worked upon. It can be used to decipher the host pathogen relationships in crops. Let's say there is a particular gene that attracts at a specific type of insect, and if you carry out gene editing in the plant, you cut that part of the gene and replace it with someone else, probably the insect will not be attracted. So pest can also be pushed away with the help of gene editing or the genome sequencing. Which can be followed by gene editing or. So yes, select the correct answers using the code given below. So as we can see, all the three options can be correct directly or indirectly. So your answer becomes D, that is 1, 2 and 3. So yes, that's all for our today's discussion. The topic is a bit heavy, but it is very interesting and it has a lot of potential. Genome India project has a lot of potential in in determining how healthy Indians are in the coming times. There are a lot of issues also associated. For example, somebody requesting you and asking you for carrying out genome sequencing of your cells or your DNA. So would you prefer that, or would you be afraid that where will this data go, where will it be used? So data privacy issues or privacy issues with regards to health, with regards to genomes, that are coming up in Indians. Plus we do not have a data privacy bill yet, data privacy act yet. So yes, those kind of issues, they're coming up, that the data might be misused or a number of issues. But yes, what happens is yet to be seen. The good part is that 2/3 of this project has been completed. 7,000 genomes have already been sequenced. So yes, stay tuned to our channel. That's all for today's discussion. I'll keep meeting you with more and more topics. Stay tuned, keep working hard, and all the very best.